Good evening. So, welcome to the fourth day of the second week. Right? Now, as it was advised and it was suggested, very good suggestion. Right? So, what I have done is, say in today's session, right, whatever the topics which we'll be covering, right. So today's topics I have included into that description, right. So and within 1 or 2 days I'll be doing it for rest of the videos also, so it will become much easier for you to figure out that what exactly is into that particular session, because that suggestion was really very good. Because when we say that, okay, this is week 2, day 1, day 2, day 3, but someone who has, say, he's watching for the first time, he or she might not be knowing that what exactly is into it, right? So from next week onwards, I'll prepare the entire list and I'll start putting it into description also, right? So that was one thing. And the second thing is regarding the chat, right? So as we start our today's topic, right, I'll actually turn the chat off, right, so that you can put complete focus on the subject. Once we have discussed everything, so at the end of the session, I'll turn the chat on. So whatever the questions which you want to ask, you can. Otherwise, you can always send a mail to me, that is at tillyourmbbs@gmail.com, right? So I'll try to reply as early as possible. Okay. Yeah, that's what I was talking about. That as we'll start the topic, right? At that point, we'll turn the chat off. And it is good. Say whatever you feel like, right? Whatever your suggestions are, just keep telling. So that's how we keep on improving. Okay. So that's the introduction. And these will be the topics for today. And after that, I'll turn the chat off and then we'll start with one more important thing, right? Say that question which was asked, right? There it became, say, it was bad on my part. I'm sorry. But when it was asked that regarding ADH, antidiuretic hormone, right? On the first lecture, I said that it acts on distal convoluted tubule and collecting ducts. And then yesterday I said it is on proximal convoluted tubule. Well, it was my mistake. Right? What really happened is that I don't know this is right. Somehow I was on Wikipedia and I was watching, and then there is a mistake in that. Right? In one bracket, instead of DCT, distal convoluted tubule, they had written PCT. And this somehow stuck in my mind, and that led to this confusion. Right? So I'm sorry for that. Right? This is incorrect and this is right. And in fact, today we'll be discussing that how actually it acts on that sodium potassium ATPase pump and what really happens, right? Which will be a very strong base for the pharmacology. So this was a small clarification, right? So please, this is correct. And I'm keeping this in our PDF also, right? I'm just saving it so that everyone knows that yes, it is the distal convoluted tubule and the collecting duct which are affected by ADH or vasopressin, right? Okay. Okay. Say, Harsh, don't worry about it. Say, I need to take the things in depth, because it really happens that, say, if the things are taken superficial, we never get 100%, right? What I mean to say is that if we decide that, okay, let's say today my target is to run 20 kilometers, definitely by 15 or 16 kilometers I'll start feeling tired. But if my target is a full marathon 42, somehow the energy comes, and up to 35 kilometers there is no problem. Same thing happens in studies also. Whatever we learn, we never absorb 100% of it, right? So, and when you watch it for the second time, immediately your brain will start deciphering that, okay, this is the point which I was forgetting. Especially when you will be appearing for those tests. As I said, that this week we shall be having 50 questions, right? So that way, even if at times, and because you are in first semester, so it is completely okay, right? That if you face any problem, if you face any difficulty in any of the point, you are free to send a mail, right? I'll try to make it more simple and send it across. Or I still suggest, instead of mail, why don't you write in the chat box? Because say, Harsh, whatever your problem is, that could be the problem of even others also, right? So that way everyone would be benefited. So okay, that is good on your part. Okay, all right. So let's start with our today's session, right? And just for our very good focus, complete focus, we shall be turning the chat off now, right? And we'll be resuming it after end of the session. Okay, right? So just give me a minute, and it has been turned off. Okay, all right. So our today's topics, as I put into the description also, but today's topics are: one, we'll be talking about insulin, right? Very interesting topics. Second would be glucagon, and our chief guest for today is adrenal gland, because we'll be talking about the entire adrenal gland, all the three zones, and this would be a real fun. Okay, so sorry, let's start with the insulin. Insulin. See, insulin is basically a peptide hormone, right? It is a peptide, and when we write H, so this means it is hormone, right? So this is the peptide hormone, and it is released by beta cells of islet of Langerhans. Yes, you need to remember this name, islet of Langerhans. This is in pancreas. Now, as we know that pancreas is like exocrine as well as endocrine, right? When we say exocrine, it means those secretions, they will be going directly via the pancreatic duct into the second part of duodenum via that ampulla of Vater, right? And that Vater is not Waiter, it is Vater, right? It is ampulla of Vater. Ampulla means it is opening, right? Ampulla of Vater. And this is into the D2. When we say D2, it means it is second part of duodenum, right? And that opens via pancreatic duct. So it will be sending those digestive juices which will be helping for the digestion. So far is for the exocrine. For the endocrine, right? For the endocrine, that's what we are talking about. There will be alpha, beta, right? All gamma, delta, epsilon. But over here we are dealing with the beta, right? So that beta cells of islet of Langerhans, they will be releasing the insulin. Now this insulin, say it passes through three stages, right? How it synthesizes. It is, and this thing will be true even for glucagon. Just the name would change. So it is pre, pro. Just remember this pre, pro, right? So then pre, pro, insulin. This is a long chain, right? So when you cut its tail, right? So tail is cut. When the tail is cut, it is called as the proinsulin. And then once again, when it is broken, then it is called as the insulin, right? So that's how it is synthesized. It is synthesized. Okay. Now, what's the main function? Main function of insulin. The main function, once again, we'll be using our same style, that this is the cell, and where is it? Yeah, here it is, right? This is what? This is blood, right? So here is the blood, here is the cell. This will be the cell membrane, right? And cell membrane is made up of bilayer of phospholipids, right? So main function of insulin would be that those glucose, right, which are playing in the blood, they should be given path to enter into the cells. But that's the whole idea, that they should be able to enter into the cells. And technically, we'll call this thing as cell uptake, right? Not a big rocket science, right? Cell uptake, very simple word. And this is by insulin. So that would be the function of insulin, what it is meant for. Insulin will do so many other things also, but this is the prime function, what it is meant for, right? So that's what it would do. We'll see it in more detail, that how exactly it will be doing this. So to see this thing, we'll keep the same figure, but this time drawing it on another page, but in more detail, right? We'll be adding something to that. So let's see, same style, right? Same style. This is the cell membrane, right? So cell membrane, and we have, sorry, we have blood over here. Okay. Now here comes the entry of insulin. Insulin tells that I need to talk to this cell, so that I should teach him how to pick up the glucose, right? So it'll say, okay, well, insulin, you are too big, you just can't enter it like this. So then no problem. I do have those receptors who can really talk to. So here is the cell membrane. He has got the receptor, right? Now this receptor is an insulin receptor, and its speciality is that it has got 4 subunits, right? This receptor has got 4 subunits. You can see parts of it. So there are 2 alpha and 2 beta subunits. So this insulin will simply come over here and will attach to this. And in the process, inside, suddenly there would be that it is called as the tyrosine kinase activity, right? Remember this word, tyrosine kinase activity. This tyrosine kinase activity is like sending an alarm in the cell, right? This is inside of the cell, right? We are this, anything which is over here, right? This is inside of the cell. Yeah, this is inside of the cell. Now this tyrosine kinase activity, right? It will send all the signals, right? It will send the signals into the cell, right? And those signals, they are going to everyone, but it goes to those who are just trapped into those small vesicles. Vesicles means they are like those balls, they are capsules, right? So they are just floating into the cell membrane, and they are having what? They are having some special structures. They are having some tubes, right? You can say some tubes. What are these tubes? They are like a substance, right? They are like an attachment which will create a path from out to in, and they are called as, we'll write it over here, they are called as the GLUT. This is glucose transporter, right? It is what is called as the glucose transporter. See how appropriate the name is, right? They will say that yes, glucose you can pass through us, right? And because there are several, so that's why we have given the number GLUT4. So all these pink color are, right? All these are GLUT4, right? These are GLUT4. So let's write the 4, number 4 on all of them, like a jersey number 4, right? All of them. So they are like GLUT4, GLUT4. Now when the signal comes, there are two things what is happening. One, it will tell, chalo, all the vesicles, you start moving on to the cell membrane, right? Go towards the cell membrane. And then over there, these GLUT4, they will start applying themselves on the cell membrane, right? They'll apply. So the final output is what? The final output is something like this, right? And those GLUT4s, right? They created these paths, right? All of them jersey number 4, right? Jersey number 4. Done, 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 right? So now this is a path which is ready. Now this path, because they fuse, technically what happens is they fuse with cell membrane. So they fuse with cell membrane, these GLUT4, right? And they have created. But this is great for those glucose, right? So those glucose which were waiting outside, immediately they'll find a path is available and enter, right? And they will enter. That's it. That's it. So this is the final output. So over here, the keywords to remember, one is the GLUT4, right? So that jersey number 4 is to be remembered, right? And that's outside. This is blood. So this is the entire meaning or the entire function of how this insulin would actually be acting, right? Now this was quite okay, but when we say in reality, then there is one important thing to know. This is what is called as the phases of insulin secretion. For us, it was so easy. Chalo, we said this is vesicle. This is one more vesicle. You want more? Yeah, here it is, here it is, here it is. It is so easy to draw. But in real life, it is not that easy, right? It will say, no, there are two phases. The first phase, the phase 1, this is fast, right? So it is for those L type. In calcium channels, there will be two types of calcium channel, right? One L type and second is the R type, right? R type of calcium channels. We'll talk about calcium channels right later, right now, because otherwise we'll deviate from the topic. But once we can understand one important thing, that this L type of calcium channel, they react very fast, and that is one point. And the second, the vesicles which are over here, right? Vesicles, those who are carrying that jersey number 4, right? That GLUT4, they are preformed, they are already there, right? Ready. So this is like a ready-made that Maggie, right? 2-minute Maggie. So immediately it is ready and ready to, so cooks fast and ready to eat. So these vesicles, they are preformed, immediately they will release the insulin, right? And done. The entire process, let me see, within 10 minutes, so everything is done. It will be continuing for say about 10 minutes and all is well, done. But well, when it comes to say this R type, so over here he said, see, secretory vesicles, those vesicles we need to form, they should be newly formed. So we have to say new formation of them is needed. Now that takes time, right? That takes time. So that's the reason that in such cases it will be slow, but it will be continuing for about say 2 to 3 hours. The importance of this, that is 10 minutes or this 2 to 3 hours, its importance is when you'll be handling the patient of diabetes, right? So sometimes when you're giving the medicine, it happens that their morning sugar is good, but then PPBS, that is 2 hours after the diet, after the food, it spikes, or the vice versa, or sometimes it is increasing in only in the night time, right? So all those variations, they are to be logically implemented using and understanding that there are insulin some are short acting, some are long acting, and then how the drugs will be acting as per their half life, its importance for this purpose, right? So how fine tuned you can really, because this is like a customized thing, right? The way one shirt won't be fitting everyone, right? So same way, one regime won't be fitting anyone. That's why sometimes it happens, like when you're treating a patient of diabetes, it takes some time because you are watching that person very carefully, that what is rising or what is falling, and then accordingly you'll make changes in the medicine, right? We'll talk about this definitely, because in pharmacology I'll be taking the subject which would be the topics which would be related to what we have already discussed, right? So next week when we'll take biochemistry, and immediately after that we'll have the pathology, and then we'll have the pharmacology. So within 5 weeks, though, you'll be very comfortable, right? Then we'll go for some very interesting studies. I wish to go for even forensics medicine. That is so good, right? Forensics medicine. It's like every day there'll be lots of stories, right? And as such, right, we all are born criminals. So when it comes to like those toxins and ammunition and guns, right? So it becomes very interesting, right? So we'll talk about all of this. And as a doctor, you should know, right? You should know that what exactly is slug. You should know what exactly is the bore of that revolver, that what exactly is the difference between that bullet, what how the bullet of that sniper works, right? It is so interesting. And what is entry wound, what is exit wound. Just by look at it, you'll be able to say, okay, this person was shot by this type of gun, right? All those things. And I got lots of images, so you'll enjoy it. Right? So we'll take that forensic medicine. Toxicology too is so good, right? Okay. And I also wish to take the psychiatry part, because psychiatry is also equally good, right? Again, it's like story. Every day it's like a story. Okay. So we were talking about the insulin part, right? So what in nutshell, that insulin is just teaching the cells for glucose uptake. True? So glucose uptake. Now think of that glucose is like money. When there is so much of money, that is when person has consumed, because glucose will come into picture. Insulin will come into picture when there is so much of glucose. So then insulin will say that, see, there is so much of extra glucose, right? If there is extra money, what we do? So let's do the FD, fixed deposit, right? So glycogen, our glycogen is like fixed deposit, and the bank is liver, right? So we go to liver and we'll say, see, there is so much of extra glucose, convert them into glycogen, right? That's our FD, fixed deposit, and deposit it, right? So when we say formation of glycogen, so it is called as glycogenesis. Genesis means formation. Lysis means to break it, right? So then this is a big bank, but then there is another small bank also, and it is in the form of skeletal muscle. So we say, okay, I will give you some deposits to you too, right? So thus glycogen is deposited into this liver and the skeletal muscle. But still we have got more glucose, right? Still, there is still more glucose. So still more glucose, because it was like that unlimited pizza, and mercilessly we consumed so many pizzas, right? Because it was unlimited, so we need to that vasulify, right? So what about that? Because so much of glucose, and this is enough, right? We have done enough FD. So now comes the process. It's like what's called as the glucose we need to convert into pyruvate. Sometimes what is done, that extra money, so chalo, let's buy something, right, which can be useful in future. So this process of glycolysis that starts, right? Glycolysis. Now in this, it starts with glucose, right? It starts with glucose. Now glucose is what? Basically glucose is 6 carbon, right? It has got 6 carbon. So this glucose is converted to glucose 6 phosphate, right? Glucose 6 phosphate. In fact, we have already talked about this in one of the session, right? Where we talked about all this thing in very much detail. But just as a recap, say then glucose 6 phosphate, it gets converted into its isomerase, right? Isomerase is what? Everything is same, just there is change in the location, right? So this is like fructose 6 phosphate. Then this fructose 6 phosphate, in that one phosphate extra is added, so at position 1, so it becomes fructose 1,6 biphosphate. But this is a very smart move, so that's why we say bisphosphate, right? So this is what really happens in it. Then, and yes, you are right that there are all those enzymes which are present in between, right? But right now, for the sake of simplicity, we are not taking it, right? Then it is broken into two parts, right? This is broken into two parts, and both are then 3 carbon, right? And this is like G6P, but name is now different. So it is glyceraldehyde 3 phosphate, right? Now 3, because now it is 3 carbon glycerol, so G3P, right? So let's write G3P, glyceraldehyde. And then other is that da, right? We said da, right? What was that da? D is always easy, it is di. H is always easy, it is hydroxy, right? Hydroxy. So dihydroxy, but rest is acetone phosphate, right? Phosphate, right? Acetone phosphate. Dihydroxy acetone phosphate. This is DHA. This is also 3 carbon. But this DHA is like it gets converted into G3P, right? And thus we have got 2 units now, 2 units of this glyceraldehyde. So we'll write just GLY, glyceraldehyde 3 phosphate. You got it? Now we have got the 2 units, because 1 unit, this DA, which converted to glycerol, and 1 to glycerol himself only. So from this point onwards, the entire process will be running twice, right? So then glycerol 3, that will be converted to 1, 3, right? 1, 3. So when we say bi, no, it is smart move, right? So it is bisphosphate glycerate, right? Glycerate. Actually, yes, you need to remember this because it is part of it, right? And then 1,3 bisphosphate, right? It will be converted to, because that's the point where those 2 ATPs will be released, right? So it will convert it to 3, but because now 3, so we have to just say phosphate, because now it is no more bi. So it is 3 phosphoglycerate, right? 3 phosphoglycerate. And then this 3 phosphoglycerate, just there is change in the location, so it becomes 2 phosphoglycerate, right? 2 phosphoglycerate. And then this 2 phosphoglycerate, it goes for that dehydrogenase, right? So that's why now it would be phosphoenol pyruvate, right? Because over here the water will be coming out. And remember, all of them are 2, 2, 2, 2, right? And finally, it is the phosphoenol pyruvate which will be converted to pyruvate, right? So this is actually pyruvate, right? This is actually all the steps, all the way it goes from, say, glucose to pyruvate. When this thing happens, right, it is from this point onwards the actual action starts. What it is? That it is like pyruvate, it tells that how is the situation, how is the condition, right? When conditions are aerobic, right, that is when sufficient oxygen is available. So it will be running what's called as the Krebs cycle, right, in anaerobic condition, because it will go for that to acetyl CoA, right, to acetyl CoA. So that's what is called as the citric acid cycle, right, citric acid cycle, or we also call it as a Krebs cycle, Krebs cycle. But let's say you are running, right? So either there is hypoxia, or we can say that anaerobic condition, right? Anaerobic condition. But still, your wish is to complete the race. You keep on running on and on and on. But now something is to be done for the energy. No problem. It does. Even in anaerobic condition, this thing continues, but the output would be the lactate, lactate or the lactic acid. And it is this lactic acid, when it gets accumulated, that is what gives to that soreness, right? And you are sweating, right? That's what really happens. It gives sore muscles, right? Sore muscles, because it is accumulated into those muscles. So that's the reason that never give up, right, when you start any of the exercise. Because when you start exercise, these things are bound to happen, because muscles are not conditioned. But if you stick to it after 72 hours, right, in 3 days, 72 hours, your body would be conditioned. And that's why this is called as the golden period, right? After 72 hours, you'll be so comfortable with any new thing you are learning. Now, this is true not only for the exercise, it is true even for the brain. Whenever you start learning any of the thing, stick to that particular topic, stick to that particular subject at least for 3 days religiously, right? Whatever happens, don't deviate. You'll find suddenly on the third day, on the fourth day, you'll find that that topic is no more that difficult, right? Your brain has adapted it to a very high level. So this is what really happens. Okay. All right. So that was like Krebs cycle, in which the final output is, say, carbon dioxide and water, and then carbon dioxide is excreted. That's how this really works. So it means that when we talk about insulin, right? When we talk about insulin, it does, right? This is we learned in our own language, but when it comes to exam, or when we have to say something, so we have to say it in bit stylish way, using some interesting words, you know. So that is when we say that it promotes, right? So, because word is good, right? It promotes glucose uptake, right? This is first. Now, I'm not writing just for the sake of writing, because I'll be asking you questions from this. So I'm putting a star, right? This time you must have noticed I'm telling you at so many points that yes, I'll be asking question, and genuinely I'll be asking. It's for the purpose because these points are so important. So once you'll remember these points, you'll be able to perform good in exam, but it will be helping you for our next subjects, right? Which will be discussing in third or fourth or fifth or sixth week. And someone asked very interestingly that what after 10 weeks? Nothing. After 10 weeks, there is 11th week, right? So we'll do something. Okay. So first is it promotes glucose uptake. The second is, as we said, that it leads to glycogenesis, right? That is glycogen production, right? Glycogenesis, right? So glycogen is what? Remember that our FD, fixed deposit, right? And that fixed deposit is done in where? It is in liver and skeletal muscles, right? So that is where the glucose is converted to glycogen, right? Okay. The third is, let me scroll the page. Yeah. The glycolysis, right? Where it will break and it will create that pyruvate. Pyruvate. Why it should be doing it? So that ATPs are formed. And ATP is a currency. right? ATP is the currency which will be utilized by everyone. So that's the purpose. That's the reason that this pyruvate is so important, because then from pyruvate, whether an aerobic or anaerobic, right, it will go in the condition as per the requirement, as per the situation, and will generate ATP. The fourth thing is there would be lipogenesis, because when it will say that there is so much of glucose, right? So we can't just say throw away the glucose. Let's convert, because who knows when we need it, right? Glucose is like a money, so we don't throw it, right? It is lipogenesis. It will say, okay, then this is like, we have to say, put this money silently somewhere, right? So that is where this acetyl CoA, right, it will go to triglycerides, right? It is these triglycerides where they will be stored, right, stored in fat. So liver will help, but then adipose tissue, fat cells, right, those adipose tissues, right, they will say, okay, we'll store, right? Not, that's the reason that when someone consumes so much, slowly and gradually you'll see that his shape starts changing, right? And the fat deposit is specific in case of females, specific in case of males, right? In males, that tummy starts increasing. In females, the deposit is into hip region and the waist region, right? So that is because there is specific affinity. But in any case, there is, say, storage stored in the form of fat. At the same time, all those amino acids, insulin will say that all skeletal muscles, right, start eating, because we have got so much of glucose, so you start utilizing it. So it will tell even amino acid uptake is also increased in skeletal muscle. The situation is that there is so much of glucose. Now here comes one very interesting part. This insulin, we will make it on another page. This insulin, it acts on sodium potassium ATPase pump also, right? Now I know you must be knowing ATPase pump very well, but still, right? I'm just taking it in brief. So here is the cell membrane, right? This cell membrane. Like one that poem, water everywhere, right? So same way over here, it is sodium everywhere. So this is the outside word. This is outside and this is inside. Right? It is inside. Outside there is so much of sodium, right? Sodium is everywhere, right? Now there is a pump, right? Now when do we implement pump? Let's say if there is heavy rain and somehow the water has come down into our home, so we'll just put a pump, and so that pump would throw away the water outside. But outside there is so much of water already, but still this pump is working against that, right? Same thing happens over here. Pump is, and that's what we say, active pump. Otherwise, if there is slope, so then no energy is needed, water will go into that direction. But if it is pump, so it will go against the resistance, right? That is what is called as the pump or the active pump, and in that case you need energy. So same thing is over here, that over here, say we draw it like this, and we say that this pump will do what? This pump, okay, outside there is so much of sodium, over here there is less sodium, right? So I'll just write it twice, but outside there is so much, so obviously the concentration outside is very high, but this pump will throw 3 sodiums outside, right, against the resistance. So it is against the concentration. So that's why this pump, we'll call it a active pump. If it is active, so it has to use ATP, right? It has to use ATP. It has to use energy. But then there is something more. And that something more is that over here, there is so much of potassium. And outside also there is some potassium. But again, against the resistance to potassium, they will be brought in. So actually what happens that there is decrease in one positive ion into the inside, and these pumps, they are running 24/7. They keep on working non-stop. They keep on working, right? So this is how it really happens. That's why collectively when we say its name is sodium potassium ATPase pump, right, which is an active pump. Insulin supports this pump, right? Now very important. See, when insulin, right, it supports this pump. So it means what insulin is doing, right? Insulin is, the final effect would be, this is blood, right? Just say this is blood outside. So what insulin is doing, it is throwing sodium in blood, right? So it is, if from the blood perspective, if you see, right, it is decreasing potassium and increasing sodium, right? Just keep in mind, right? Because when we'll be treating this a diabetic patient, these things will come into picture, right? Why? Because there is either increase or decrease in this insulin, so it will be affecting sodium potassium pump, and that's how the ions, they will be affected, right? So at that point we'll be talking about this in more detail, but just keep in mind, right, this is the point, that as the sodium is getting pushed into the blood against the resistance, so there will be increase in sodium and decrease in potassium, and everything what we are talking about is with respect to blood, right? Just this much. Okay. Now when we are doing this, right, insulin will say that when we have got so much of glucose, so what insulin will stop or will not do, right? So in other words, what we are telling is, in books, when we say that insulin will inhibit, right? This is like a very sharif sentence, right? Technically, what we see are, there is so much of glucose, right? When we have got so much of money, right? We are rich. So then will we break our FDs, right? No, right? There is money to say to spend. So then will we do that? Nahi yaar, kuch bhi bech ke get the money. No, we have, right? So same thing happens over here. Remember, there is so much of glucose, right? So then insulin will inhibit what? Insulin will inhibit, say, that glycogen, you remember? Glycogen, which was our FD, right? Will we break our FD if we have got enough money? No, we won't break. So then glycogenolysis. Lysis means breaking the FD of glycogen, right? No, will not break. So this is what would be inhibited. Glycogenolysis will not occur, right? So this is first thing. Second, right, if we need money, so then we say, "Kare yaar, just give me some." We talk to one friend, we talk to second friend, right? From any of the source, we just get the money, right? Especially if it is, say, 14th of February, right? I think it's Valentine Day, right? Just giving you idea, okay? But we see that, nahi yaar, aaj to paisa chahiye, right? So for that, we try all our resources. This is what is called as the, say, this glycogenolysis was a decent way. This is It is not indecent, but new resources. So that's why it is called as the gluconeo. Neo means new, right? Genesis, again formation. Are there is enough money. Why do we really want to create more money, right? From other resources. So this is what is called as from non-carbohydrate, non-CHO is carbohydrate, right? Non-carbohydrate sources when the glucose is created. No, we have enough glucose, we don't want this also. So that means insulin will inhibit glycogenolysis, that is FD won't be broken, and insulin will inhibit gluconeogenesis, right? Because we don't want to create more glucose even from other resources, right? Non-carbohydrate resources. So right now things are happy. So that's why these two things will be inhibited. What else? As we said that when what glucose was doing, I'm sorry, what insulin was doing, insulin was even telling that, okay, let's deposit something in the form of those adipose tissues also, right? That, so over here, lipogenesis, right? So even there it was depositing. It will say, no, no, no. This ulta is lipolysis. So why to trouble even fat cells, right? We don't want to break them. So no lipolysis, right? No breakage of those fats. So fats are not to be broken. Okay. What next? Now only the protein is left out. So should we break protein to get some energy? Array, why? There is enough energy. So over here, this we won't be breaking even the proteolysis, right? So proteolysis, even that won't be entertained. No need, right? So over here, so no breakage of fat or protein. So no lipolysis, no proteolysis, nothing, right? Everything is good at this point of time. Okay. So when there is increased glucose, right? When there is increased glucose, increase means beyond limit, right? So then it is called as the diabetes, right? Diabetes. In these 10 weeks, when we'll be talking about medicine, yes, this is the topic which will pick up, right? Or you can suggest any other topic, but at least on one day, we'll definitely be taking the diabetes. So quite basic, right? It is, there is one what is called as the type 1, and second is what is called as the type 2, right? Type 1 diabetes and the type 2 diabetes. Now this type 1, that is a bit painful, because when you look at those patients, they all are cute little babies, right? Now these diabetes, they are having to those patients when there is genetic abnormality, right? Genetic abnormality. Genetic abnormality. So in which it happens that T cells of our own body, they start attacking those beta cells. And beta cells are when affected, so that would lead to decreased insulin. And once the insulin is decreased, so now there won't be any FD, there won't be, say, any of the deposits into any of the liver or skeletal muscle, and then that money will keep on moving into the blood, that is increased blood glucose. Now this increased blood glucose, is it bad? Yeah, exactly. If we don't deposit the money into the bank and we keep it in our pocket, we keep on spending lavishly. Same thing happens. And in that case, over here, even those bacterias, right, they'll start, because glucose is the world's best culture media. So that minor infection, which could have easily be subdued, but now because of the presence of that glucose, because this glucose will be consumed by all those bacteria, and thus the infection will go uncontrolled, right? This is a very simple procedure that when it comes to diabetic patient, you'll find that healing is slowed down, right? Such a slow healing. It is because this glucose is feeding those villains, right? So that's what really happens. In case of type 2, should not happen to anyone, right? This is like a lifestyle disease. It's like a lifestyle disease. So over here, maybe because of obesity, lack of exercise, right? It leads to what's called as the insulin resistance, right? Insulin resistance. Now, as you know the procedure, you just learned, right? Insulin resistance, that is by the cells, right? Or in the cells. Now, what really happens? You remember, this was the cell, right? When the insulin comes, right? Here is its receptor, alpha, alpha, beta, beta, right? So that insulin comes over here and it should act, it should lead to that intracellular signals. What if these cells, they say that, well, we won't respond to this one insulin he is coming. So he'll say, pancreas would say, Acha, I'll send more insulins. So now there are more insulins. So then now this cell would say, okay, chalo, now I'll respond, right? So previously, those channels, those Jersey number 4, those GLUT4, which were getting generated just by one call, now they are getting generated by 3 calls. It means it will be putting load on pancreas, right? It will be putting load on pancreas. Why this cell should be resistant? It is because of constant firing of insulin. Why insulin is coming again and again? Because we are eating so much, right? That one movie, and then that movie will be lasting for 2.5 hours, and within that 2.5 hours, right, we are eating constantly, even without knowing that what we are eating, right? That entire Lay's chips packet, right? Why to have small packet, big packet, right? It is khatam, right? Then we eat Coke, this, that, everything, right? And then at the end, if we just make a total of number of calories, it is skyrocket. But inside story is that insulin, he is just watching, are still kha raha hai, chalo. Then blood glucose is spiking, put more insulin, put more insulin, put more insulin. And then exactly what happens to this cell, right? He tells that you are constantly telling me, ja, now I don't follow you, right? It is exactly like that. If someone keeps on telling, अरे चलो पढ़ने बैठो, then brain tells that, yeah boss, I'll do everything except this पढ़ना, right? That's how our brain works. So same thing, these cells, they say you are telling constantly, now I'll not follow with that small dose. Up to, if there are more insulin, then and then I'll do it. The load is on pancreas, and one day pancreatic cells, those beta cells, they'll say, okay, boss, we are retiring, enough of the work has been done, bye bye. And that's what is called as the overload. It is at that point that these beta cells, they start atrophying, right? They atrophy, right? And when they die, so obviously there is decreased insulin, right? Already the cells are having so much of resistance. There is no option but to give the external insulin, right? That's why it is what is called as the lifestyle disease, right? So do remember, and that's why it is said that not more, just 20 minutes of exercise. And what do we mean by exercise? Right? So we are distracting slightly from the topic, but doesn't matter. This is like 220 minus your age, right? Let's say your age is 20 years. So it comes to 200, right? A rough, right? There are some complex calculations, but this is a very rough calculation. Even if you do this much, it's more than enough. 80% of your heart rate, right? This is like 200 minus 80, so it comes to 160, right? If you do anything, whether you want to do skipping, running, whatever you like, right? Keep your heart rate from 140 to 160, right? For 18 to 20 minutes, and then the cool down. Remember, for the stretching and warm up, and then the cool down, right? 2 minutes each, half an hour maximum. It will keep you fit for rest of the life. And again, I'm telling, this is like an investment. Any investment, if you do at this young age, when you'll be at the age of 50, you'll find you'll relish the fruits of it, because then that thing would have multiplied so much. Same way it is for fitness, right? When you do it right now, because you are in the best state of your life where the energy level is maximum. So that's why you'll be performing much better. And your muscles, that muscle power will help you later in life, right? Okay. So our first topic was insulin. Now the second one, what we said about the glucagon, right? Deliberately I wrote it like this, right? Because it is, someone must be very creative to give this name, right? Because this glucagon is when the glucose are gone. So at that point, the glucagon will come into picture, right? So that's why it is like glucagon, right? So here it is, as we were telling about, say, these pancreas, right? Exocrine, endocrine, right? Exocrine, endocrine. Exo means it is always via duct. So over here, pancreatic duct, and then there were like all those digestive juices, right? It goes via that pancreatic duct and opens into the D2, right? Correct. That is the second part of duodenum. This endocrine, which drains in blood, right? Its secretions, they are put it into blood. So over here we were having alpha, beta, gamma, delta, and epsilon. I liked one thing very much, because say some of the senior students, they are answering the questions in that chat. Actually, I tell you, it helps me so much, because there are mails, so many mails, then there are few reply. I try to reply almost everyone, but this help I really appreciate. Someone called Mandar, right? He replied, so really thankful to him. Okay, so here it is alpha, beta, gamma, delta, and epsilon. I remembered this because I think her name was Preeti. Yeah. So she asked that what is this epsilon, right? So he replied that okay, it is this ghrelin. So that way, so when you start helping each other, that is also a very good thing. Okay, so we now know about this beta, that is insulin, right? That we know. So this, when the glucose are gone, that is the glucagon, right? That is by alpha. This gamma, it is for those pancreatic peptide, right? Those hormones. This delta, right? It is somatostatin. This somatostatin is like a reset switch, because it can stop everyone, right? It can stop insulin, glucagon, GI motility, everything. We'll talk about it, right? Though this was not the topic of today, but yes, we'll be discussing immediately after glucagon, because it is important. And this epsilon, it is that when the stomach, when the stomach is empty, right? So literally it's like rats are running in the stomach. So then how is that sound, right? When you are very hungry, it's like grrr, right? So that's why here also, see, the medicos, they are so creative. That's why they must have given this name ghrelin, right? So that is ghrelin. So this ghrelin is like hunger hormone, right? It is like hunger hormone, right? When the stomach is shouting, right? So when the stomach is shouting, lao lao, right? So that is ghrelin. So now we will be focusing on this glucagon. So for glucagon, again, same thing, right? Pre, pro. I said you just remember pre, pro, pre, pro. Previously it was insulin, so now it is glucagon. Glucagon, right? So how it is synthesized? This is pre-pro glucagon. That is the beginning. right? So this is, all these are single peptide, right? Single peptide. It's a long chain. So when the tail is cut, right? Right, when the tail is cut, so this becomes proglucagon. And then once again, when it is chopped, right, literally chopped, so many pieces, and then there is formation of glucagon. And then this glucagon will be going everywhere and will be functioning. But this glucagon, right, into the cell, it is trapped, right? It is packed, right? It is packed into those granules. So in the cells, in the form of those granules, right? This is, so we'll just write G, no, that will confuse with glucose, right? So let's, this glucagon. So we just write like this. These are all glucagons, right? They are packed into those vesicles and they are waiting, right? They are waiting for the signal. Then as soon as they say, yes, glucose are gone, so that is a message for all of them, and then they will rush, right? And then they'll start acting. This is, in fact, a very easy topic, glucagon, because it would be just the reverse of insulin. Almost at every point, it would be just the reverse, right? So, as we said, that the first point is that the alpha cells, right? They are responsible. Now, these alpha cells, they are sensitive to glucose, right? They are sensitive to glucose. So, right from the beginning, they'll start sending, as a glucose level is dropping, right? So we need to send this our soldiers, right? These glucose are gone, glucagon into the blood, right? So they are sensitive to glucose levels, right? So decreased glucose level will lead to, say, this glucagon in blood. How? We'll just see, right? Second, what this will do, it will increase blood sugar level, blood glucose level, right? Now, when do we need more glucose, right? When we want to hit someone, when we want to fight, right? So whenever we want to fight, we need energy, because then and then there has to be a power in our punch, right? So the system is sympathetic system. Sympathetic nervous system. Now this sympathetic nervous system will what? It will lead to that secretion, adrenaline, right? So this adrenaline will come to this glucagon. Let's see, we are about to go for that fight. We need energy. So you start working, right? So you'll say, okay, good, right? So these are the two prime things. Third is, there is a hormone called CCK, right? You remember this? Cholecystokinin, right? This cholecystokinin, which has got so many GIT functions, like say it regulates gastric acid secretion, right? Motility, right? Gastric acid secretion, etc. But it has got one very interesting function. It goes and it tells those pancreas, right, that you go for those, it stimulates those cell growth in pancreas, right? So this is stimulation, right? It stimulates cell growth in pancreas, and that is when the cell growth is there, so obviously that will increase this glucagon level, right? So that is where increase glucagon. But well, the cell growth is increased. So will it increase insulin also? Yes, it will, right? It will. So it will increase insulin as well as the glucagon. So that's why it is just a minor point where it tells that yes, this is what it will do. But actual control, that depends upon the situation. One very important point is, when you genuinely need more energy in the form of exercise. That's where that's where the glucagon will come into picture, because you are working hard, so that's why you need more energy, right? Now it's like sometimes, say, in those marathons, sometimes Red Bull is given, right? Now is it good, Red Bull? Well, at that point, when you are tired, when the brain is tired, because it is the continuous running, right? Since 3 hours, 4 hours, right? In that heat you are running, situation is bad, right? There are blisters in your feet, and you are so tired, so that caffeine would help for some time at that point of time. But then it is loaded with sugar, loaded with sugar. So suddenly there'll be high amount of sugar which will be pushed into your body, right? High amount of sugar. What happens is that insulin, that pancreas will say, what is this, right? Instantly, as there is large amount of sugar, large amount of insulin will be pumped in, and you know what insulin will do. So insulin will say, Kare, this much of faltu glucose, chalo, let's start going for those fixed deposits, right? So it will start pumping it into liver. So initially, when you take such things, it will give a strong surge, but then suddenly there will be hypoglycemia. So take care of such things when you are going for a long distance running, because it's not like a sprint, right? Sprint is like that for short distance. Yes, you go for such thing, it is okay, but whenever you go for any of such drinks, right, there is very big spikes into the form of, say, this insulin and glucagon, and any of such spikes, it is dangerous for the body, right? Okay, so over here, the glucagon, right, this glucagon is stopped by whom or inhibited by whom, right? It is inhibited or stopped by one simple thing: when the glucose level is high, right? So high blood glucose level will definitely stop the glucagon, right? And second is, this is the point, by somatostatin, right? As we said, that this by somatostatin. So this somatostatin, they inhibit hormones. Now, it's not so. It avoids actually excessive release of pancreatic hormone. So it will stop insulin also, it will stop glucagon also, everyone. So this is like a control, right? It is like a control. So it avoids excess secretions, right? So that's what it really does. Now let's see how this glucose and how this glucagon will be acting on the cell, right? So let's draw it on another page. Same style, right? When this glucagon is coming, right? This is glucagon is coming from the blood, right? Because it is endocrine, released into the blood. Now over here, the receptor is what is called as the 7-pass receptor, right? So it goes one, let's change the color. 1, 2, 3, 4, 5, 6, 7. As it is passing through the membrane, it is also called as the 7-pass receptor, or it is called as the transmembrane receptor, right? It is called as the transmembrane receptor. It is also called as the integrated receptor, right? All those mean the same. Integrated means that if you really try to take this receptor out, you have to break the cell wall, right? It is integrated, a part of it. It cannot be separated, right? So that is what is called as the 7-pass receptor. They are also called as the GPCR, G protein coupled receptor, right? That is also, it is same. So G protein coupled receptor or 7-pass receptor or transmembrane or integrated, practically they all mean the same, right? Okay, so here is the glucagon, right? When it attaches, or actually it activates intracellular proteins, right? Now this is a hormone. This is glucagon is something which will destroy, which will cut, right? So that's why this is catabolic. This catabolic hormone will cut down something. So what it will do in liver, for our understanding, it will say that that let's break that FD, right? That FD was what? It was glycogen, right? So that was our FD. Let's break it and get the glucose. So technically, what name should we give it to? We'll say glycogen. So that is name of our FD. O, right? Lysis, break, right? So we say OFD break. So that is glycogenolysis. It is big. There is shortage of money, right? There is shortage of money, and it is 14th February, right? We need to have some money, right? So for that special occasion, let's call our friends, right? Kahin se bhi those new resources. That is gluconeogenesis, right? This is gluconeogenesis. So what was that? Glucose from non-carbohydrate sources, right? So that is gluconeogenesis. Yes, this is also needed. This is also needed. Right? Anything else, right? So we say that yes, what was that fat doing, right? So why not to ask him, right? Our old friend, right? आज तो मांग सकते हैं उसके पास से पैसा. So it is like lipolysis, right? So that's where we ask that fat. It can something be arranged, right? So he'll give something. So that's the output would be the fatty acids. And these fatty acids are what? These fatty acids are nothing but the non-carbohydrate resources. So we'll say, Acha, let's use it. So this gluconeogenesis is still triggered because this fatty acid will be acting like a raw material for this gluconeogenesis, right? So that's how the entire game of this glucose, right? The balance is like, it's like a tug of war. The insulin is on one side and this, when the glucose is gone, right? So when the glucose is gone, it is glucagon. Glucose are gone, right? So it is glucagon. That's the tug of war which keeps on happening between both of them. This is by beta cells and this is by alpha cells, right? So this was about the glucose, that is insulin and glucagon. Now we talk about adrenal gland. Somatostatin, right? We were talking about somatostatin. It is important. It is by that delta cells, right? Delta cells. And as we said that it is a sort of a reset switch, right? Again, it is a peptide hormone because all of them are peptide hormones, right? Its main function would be just to stop everyone, and make the entire system silent. Then once again things will start working up, right? So its main purpose, right? We can say that what's the purpose of this somatostatin? Name itself is taking that it is static. It will keep everything on halt, right? So that's how we remember somatostatic. It is statin, but it's okay to say somatostatic. Static means halt. So it will say, okay, halt, stop, insulin, right? Stop, glucagon, right? Not only this much, it will say even the pancreas has got the exocrine function. Stop that also. Stop pancreatic exocrine secretion, right? Even that also, yes, stop that also. Then stop all those CCK, gastrin, all that is inhibit GIT hormones, right? Not only that, he'll say that statue to GIT, right? Muscles. So GIT motility, right? They will be, that would be decreased, right? Even they are not allowed. Blood flow, decrease that, right? Blood flow is also decreased. So this is what really happens. Somatostatin actually puts the entire system into that static mode, stop, right? And that's how we say that it is like a reset switch. It's like a reset switch. And how can he really do this thing, right? How can he really? So this somatostatin, right? Somatostatin, it is in blood, right? Here is our cell wall, right? And because it is GPCR 3, 4, 5, 6, 7, right? So this is like 7 pass transmembrane integrated G protein coupled receptor, right? So this is GPCR, and this somatostatin will come over here, right? It will say, chalo, let's do some intracellular activity, but because he is Gunda, right? So he'll say its activity is like that. So he'll say, no, 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 I'm not over here to stimulate, right? He'll go and will say, stop that adenyl cyclase, right? Adenyl cyclase. This adenyl cyclase, right? He takes his cycle and then he starts doing so many activities. Stop that. So when it is stopped, so this will lead to inhibition, or we can say decrease in cyclic AMP. When the cyclic AMP is decreased, it will lead to decreased calcium. Now, when the calcium is decreased, obviously everything will come to an halt, right? This is what actually it does. So thus it has got the inhibitory or the reset system like effect, inhibitory effect, right? That's what is done by somatostatin. So whenever there is any fluctuation in glucose, when you consume glucose, fatty acids, right, amino acids, so all it this will come into picture. Okay, so that's how this somatostatin works. The next is, now we go for our guest of honor, adrenal gland. Now this adrenal gland will be comprising of so many names that whenever there is any fight, you give that name to anyone and then he won't be able to answer, because such long interesting names will come of all those compounds, right? So we'll try to remember, and in fact you have to remember just this thing, right? What is this? This is a glass, right? And there is water, right? So whenever there is glass, so we'll say hydroxy. It means it looks like that water is given. And when we'll be drawing this symbol, this, it means it is dehydroxy, means we are not giving water, right? And actually the all the processes, they will occur like this. Once they will give water, no, once they will not give water, and then obviously if someone is not giving us water, we feel so bad, then he'll say, okay, take 1 glass, okay, shall take 2 glasses of water, right? That's how these systems, they'll keep on working. So adrenal gland, right, which is sitting on the kidneys like a topi, right? So it is that's why it is called as the suprarenal, and the right side and the left side. So right side is adventurous, right, going to Egypt many a times. So that's why it is pyramid shaped, right? It is pyramid shaped. And some very creative person amongst you, right, she said that L is for love, right? And that's why that is that half moon shaped, right? So that is the shape of the suprarenal. Well, that's right. And this is the crescentic, right? It is called as the crescent shaped or the half moon shaped. Well, quite creative. Okay. In this adrenal gland, we say outer cortex and inner medulla, right? Always speak as if it is a music. Outer cortex and inner medulla. Right? Medulla. Medulla is in middle, right? So we never make mistake in that. Okay. Now comes the part. This is, though we have already talked about it, but just as a small refresher, the things start from hypothalamus, right? From hypothalamus, things go to pituitary, right? In pituitary, it is, there are two parts, anterior and posterior. Posterior, well, posterior is about that neuro. This is anterior, right? This is anterior. That's why it is adeno. Adeno means something which will be secreting, like it will be behaving like a gland. Glandular. In glandular, there were two types, right? One was acid, one was base, right? Acidophilic and basophilic, right? So one was acidophilic, one was basophilic. Basophilic, we remember like fat, right? So this was FSH and LH, right? Then this was TSH, and here is our today's guest, right? That is ACTH. That's what we shall be talking about. It means this basophilic. On histology, these cells, they will be staining blue, right? And you remember I talked about it, that for males red is red, but for female they say they are so idiot they don't understand the difference between red. This is not red, idiot, this is maroon. But for males red is red, right? All 20 shades of red are red only. So over here, that's why over here, for us, blue is blue, purple is also blue, everything is blue, but do remember, right, that it looks like either purple or blue. This is the basophilic thing. So FSH, LH, ACTH and TSH. Just over here, rest of them, right, that is the growth hormone and prolactin, they are all acidic. Okay, so here we are dealing with anterior pituitary. So over here, hypothalamus will say corticotropin releasing hormone. It will go to anterior pituitary. Anterior pituitary will release ACTH, right? ACTH, adrenocorticotrophic hormone, right? And that will travel all the way to adrenal gland. And in adrenal gland, it will act and the action would start, right? Just to be, say, drawing like this. Again, this is like 7 pass receptors. So 1, 2, 3, 4, 5, 6, 7. 7 pass receptor. This ACTH will come over here, will sit, and here is the action, right? This action will be occurring in the adrenal cortex. Cortex is outside, right? So adrenal gland over here, right? It is the adrenal cortex where the entire game would be played. Now this adrenal cortex has got 3 stadiums, right? So GFR or the layers, they are called as the zone or the zona, right? So it looks cute, so that's why zona. So glomerularis, right? Zona fasciculata and zona reticularis. So these are the 3 zones, and it will, all the 3 games will be played onto these 3 grounds. But interestingly, one very nice thing is that there would be only one thing, that is cholesterol, right? Do remember cholesterol. It is this cholesterol which will be utilized as a raw material for playing all the games. So this cholesterol is where it is into the blood. So from blood, this it will enter into this zona, right? And then the games would start. Remember that 7 pass receptor, right? That is ACTH. ACTH, though this cholesterol is into the blood, right? It is into the blood. But this cholesterol has got no class to enter into this adrenal cortex, right? Because it is controlled by ACTH. It is that ACTH will say that, okay, now you enter, only then this cholesterol will be able to enter into this region, right? So first thing first, what ACTH would do, let's write it here, right? Purposely I'm writing it over here. So what this ACTH would do, ACTH will stimulate, right? And his name is cholesterol desmolase. ACE means enzyme, right? So cholesterol desmolase is enzyme. So this cholesterol desmolase, he will act on that cholesterol, right? I'll just write for the sake of simplicity, cholesterol. I know it was here, so right, same thing, this. So it will act on that cholesterol. When it will act on cholesterol, the effect, what will do? This cholesterol, right? I'm writing again over here. This cholesterol will be converted into, right? Will be converted into pregnenolone. Pregnenolone. I used to forget this, right? This pregnenolone. Then I remembered it like that in pregnancy, female is never alone, right? So that's how I remembered. So I'm writing it over here. So in pregnancy, she is no alone, right? There is someone always with her. So then it became easy to remember. Pregnenolone. So now it is okay, right? This pregnenolone is vital because this is like a precursor. Precursor is raw material, right? This is the raw material which will be used by everyone, all those layers. Now interestingly, so actually they are like those three stadiums at various levels. So one will produce something, will send it to second, second will produce something, will send it to third, and that will keep on running, right? So it is like raw material for all three layers, all three layers, right? And those three layers were glomerulosa, fasciculata, and reticularis. So this glomerulosa will be creating those mineralocorticoids, right? Which will be affecting mineralocorticoids, which will be affecting all the ions, right? Then zona fasciculata, that will be affecting and that will be releasing glucocorticoids, right? Very important function. And reticularis will be releasing all those, say, sex hormones, right? Or androgens. This pregnenolone, it will be like a raw material for all of them, right? So let's start with the layer 1, that is zona glomerulosa. Just one pregnenolone, right? Pregnenolone. This is converted to progesterone. But from this point onwards, all names are easy. Progesterone. Now, who will do this? It is 3 beta, because 3 words will come, right? H S D. Let's start guessing. H, always hydroxy, right? It is always hydroxy. So this is easy. S is steroid. right? S is steroid. Okay. D is dehydrogenase, right? Dehydrogenase. Name itself is so brutal. Dehydrogenase. It is like someone is not giving us water, right? So let's draw this, right? Dehydrogenase. No water. So that's the situation. No problems, right? This progesterone. Now this is like a roller coaster ride, right? So be ready. This is where it will be converted to, right? Just a minute. Let's make it big. Yes, this is good. So this progesterone will be converted to, say, 11 doctors, right? And that is by giving them some water. Right? See, previously no water was given. Now here the water will be given, right? So 21 hydroxylase, hydroxy, right? Lase is, so will always come by default because they are like enzymes. Here also it is because it is enzyme. But now this is 11 doc, right? What is that? 11. D is D. Yeah, D is D, right? O is oxy. And this C is corticosterone. Because this corticosterone word will keep on coming again and again, right? So corticosterone. This corticosterone, right? I'm just telling you that he is somehow related to Sylvester Stallone, right? So that's why they have kept this name quite similar. So corticosterone, right? Cousin of Sylvester Stallone. Okay. So this corticosterone, right? This 11-deoxycorticosterone, again, one roller coaster ride, is converted to best. That's what we underlined. So it is corticosterone. Right? So this is easy. And who is doing this? One more glass of water. Right? So this means he is hydroxylase. That is so definite. So let's write this hydroxylase. So this is happy thing. Okay. Then we need to add something to it. Well, we are dealing with 11 doctors. So let's put it 11. Right? And beta. Right? So 11B beta hydroxylase, it converts 11-deoxycorticosterone into corticosterone. But all the difficult portion is complete because from this point onwards, it is a very smooth, easy ride because corticosterone will be converted to our final product, which was aldosterone, right? Aldosterone. And if you have understood till this point of time, all the three zones now are a piece of cake, right? Absolutely easy. So aldosterone, right? Who will be converting it? So it is like synthesis of aldosterone, right? So we'll write it that synthesis. Synthesis. Synthesis means production. So synthesis of aldosterone. So let's give a stylish name, right? So it is like aldosterone, right? And he has to be an enzyme. So give synthase, right? Synthase. So aldosterone synthase. But that's it, done. This was the final product, right? What was needed. And give a group name mineralocorticoids. So mineralocorticoids. So done. This is the output, right? Now, interestingly, we talked about that renin-angiotensin system, right? Renin-angiotensin system, something with that juxtaglomerular apparatus, and then in that there was like angiotensin 2. So then these are the lungs, right? These are the lungs, and those lungs, they were releasing angiotensin 2, right? Because two lungs. right? That's how we remembered two lungs, right? So they angiotensin 2, and that angiotensin 2 will be stimulating this aldosterone synthase, so that corticosterone can be converted to aldosterone, right? And in which condition this angiotensin 2 was getting released? It was getting released whenever there was drop in the blood pressure, right? You remember there was drop in the blood pressure, maybe because of burns, maybe because of some hemorrhage, right? So when there is decreased blood volume, because they all are sensitive to it, so when there is decreased blood volume, so that means there would be decreased blood pressure. So at that point this angiotensin 2 will come into picture. So why so? Why such an odd thing, right? Now see the beauty. This is super exciting, right? This portion at the end you'll say, wow, right? What a system. Now see what it will do. This aldosterone synthase, right? You are stimulating it, right? So now this is stimulated. He'll do happily the conversion of corticosterone into aldosterone. So okay. So sorry. So far good. Now this aldosterone will be having some work on, say, this sodium, right? What it will do, right? So this sodium, I'm just writing sodium over here, right? So that there is continuation, just like this. Okay. So now we have got the sodium. This sodium will be, say, this was what, that is our kidney, right? In kidney, distal convoluted tubule and the collecting ducts. This is the target area, right? Over here, at this target area, when the aldosterone comes, right? When this aldosterone comes, and this aldosterone will act on this area, and it, in this region, it would stimulate the action of sodium, potassium, ATPase pump. Okay. Now we know about sodium, potassium, ATPase pump. Let's draw one new figure. So here is our cell membrane, right? As always, this is a cell membrane, but this time this is that tube, right? So that's why I've drawn this. So this is what, this is a tube, right? It is a tube, right? Distal convoluted tubule or collecting duct, whatever, but it is a tube. Outside the tube, what is there? That environment is blood. Okay, this is blood. And then there is a sodium potassium pump. Yes, there is sodium potassium pump, right? This sodium potassium pump, this sodium potassium ATPase pump, this pump has been made active, right, overactive, by the stimulation of aldosterone. So aldosterone said that, okay, start working, right, effectively. Now it is working. Over here, it will do what? First thing, as it is said, it will push the sodium out, right? So sodium is out, 3 sodiums out, and those 2 potassiums, they are in. Now this area, right, it is what? It is what will go into the urine, right? It will go into the urine. And over here, this sodium is coming out. Now remember one golden principle. Water always follows the sodium, right? Water follows sodium. So sodium is going out. Along with that, water will also trickle out, right? Water will also will come out. It's the water. So when the water is coming out into the blood, right, so it will increase blood pressure, correct? Because it is increasing blood volume, right? See the system. Those lungs, they felt that was blood pressure is falling, there is less volume. So let me go and tell this aldosterone synthesis by this angiotensin 2. So angiotensin 2 will go and will pin aldosterone synthesis ke boss kaam karo, right? So he'll say okay, sure, right? And corticosterone will be converted to aldosterone, and then that aldosterone, he goes and he tells this DCT and CD that where is that sodium potassium pump? They say ke ye raha, right? And he'll say ke chalo start working more. Done. And in that process, when this is working, sodium will be pushed out. Sodium is pushed out to like a tail. Water will also go along with sodium. And now in the blood, there is more blood volume. There is increased blood pressure. Problem is solved. Right? How interesting. Right? This is what really happens, and this is the beauty of that mineralocorticoid. So whenever there is increase in blood pressure in pharmacology, same principles will be implemented, same principles, and you'll find that pharmacology is so interesting, right? I won't say that it is like easy, but it is easy to understand if we remember this, right? So then it becomes very interesting, and when we learn it in this way, you'll never forget. And if you have understood this first portion, right, trust me, rest of the second and third, zona fasciculata and reticularis, within minutes, you'll be able to finish it. Within minutes, right? Because most of the things, they are like this roller coaster with similar names, only some changes, right? So here it is, says zona fasciculata. And it is fascinating, right? Same, say, pregne, no, loan, correct? Because I am writing it like this because that's how I remembered. And I felt like that no, if I write it like say this, like say pregne, no, loan, how complicated it looks like, right? So that's why I avoided writing like that. Because see, these are the notes which you'll be utilizing. So when you will see this, it will flash in your brain and you will never forget it, right? Okay. It converts into what? That easy word progesterone, you just can't miss it, right? Progesterone. And then it is done by whom? That 3 beta HSD. है, and this same thing, same, no new things are coming up, right? Over here, H was what? Hydroxy, right? Hydroxy. S, steroid. And D, I'll not tell you, right? It was dehydro. Yes, you must have spoken also. I know you. Dehydrogenase, right? Is to we write because it is an enzyme. So this is done, right? This entire thing, interestingly, it is happening in zona glomerulosa, right? It was happening in glomerulosa. It comes to fasciculata directly. So this first step, right, it is getting done in glomerulosa. So this progesterone, it enters into fasciculata. So that means now the game would start from this point onwards, right? So here is now the game would start. Now over here, it happens like pregnenolone and the progesterone, both of them would come. Progesterone, which was converted from pregnenolone to progesterone in the glomerulosa. Now both the material they are coming. right? When they come, there is one 17 alpha and so you know now who is hydroxy. Hydroxy. And yes, is hydroxylase. So this 17 alpha hydroxylase will act on both of them. So this is good. Now there is just one name which we have to remember. And this is now 17 alpha hydroxy, right? Everything will be hydroxy and then this add pregnenolone. So pregnenolone. This is the output. And how about progesterone? Exactly the same. 17 alpha hydroxy and progesterone. Correct? Progesterone. Now at this point, I am telling you one very vital thing, right? Remember, not for question, right? Not for exam question. This is for that for zona reticularis. This product will be taken into the third layer, right? So over there won't be any pregnenolone, there won't be any progesterone, but this 17 alpha hydroxy pregnenolone and 17 alpha hydroxy progesterone, they will be taken to third layer. So that's why I have put this, right? I'm writing 3 also. That is the third layer, right? That is the third layer. And we'll draw the same symbol into the third one. So you'll always understand it. Okay. One interesting thing happens. Now, why to remember these two names, right? Why to remember? So there comes one smart move. He tells that finally, so it is the 17 alpha hydroxy progesterone which is working, right? So let's convert it. So this is 3 beta HSD. Now, so you know this 3 beta HSD so well, right? You know this, right? So 3 beta HSD, right? Try to remember what was it, right? It was correct. So we know this part. It is dehydrogenase. Chalo, that is done, right? A is because it is enzyme. So what was H? Hydroxy, and what was this S, right? It was steroid. So it is also easy. So now suddenly these things are not that difficult, right? And every time it happens like this, glass of water is given initially and then they take it away, right? So that doesn't matter. So this is hydroxysteroid dehydrogenase, which will convert 17 alpha hydroxy pregnenolone to 17 alpha hydroxy progesterone, right? Moving on to the next one. Now we have got 17 alpha hydroxy progesterone. So this alpha hydroxy progesterone, I'll write it over here. 17 alpha hydroxy progesterone, right? Hydroxy progesterone. It would be converted to, but final stage has come, right? 11 doc. Yeah. But over here, it is 11D, yes, oxy, but this time, last time it was cortisone, right? This is cortisol. Cortisol, that's what we have to remember. 11-deoxycortisol. And that is by whom? 21 hydroxylase, right? Xylase. 21 hydroxylase. This 11-deoxycortisol will now be converted. Now that is the last ride, right? It is converted to, see, cortisol is underlined, so it converts to cortisol. Done. Journey is complete, right? And then how about it? Giving one more glass of water. This is 11 beta. Same thing. It was in previous layer also, it was the same. Hydroxylase. So the only change, only the new name which has come up is cortisol, where it was cortisone, right? But that's it. Now this cortisol is created. This cortisol will be acting on cortisol, right? It is acting on that sympathetic nervous system. Now it is easy. Sympathetic nervous system, and will lead to everything like, say, physical stress, right? Or emotional stress, any of the stress, that is what is handled by sympathetic nervous system. So it will say that we need to fight, right? Because it is fight and fright, everything. So whatever. So we need to fight. When we need to fight, we need what? We need glucose, right? We need glucose. We need energy. When the glucose is needed, we need to produce the glucose, right? But it will say there is no time. This is for fight. So whatever the resources are available, make the glucose. So you'll say, now this is not the official way to get the glucose. So that's why let's go for this newer method, neogenesis, right? Because these are the methods which are like non-carbohydrate sources. So what are the non-carbohydrate sources? They say they are amino acids, they are non-carbohydrate. Okay. Then fatty acids, they are non-carbohydrate, right? They will be giving instant energy by the process of gluconeogenesis, right? Gluconeogenesis. Okay. Then what this will do? It will do one more thing, this cortisol. It will go to proteins, right? And it will break them. When the proteins, where is it done? It is in muscles, right? It is in muscles. When it is broken, it leads to what? It leads to amino acid, right? Okay. And one more place, it will say, what about fat? Let's break it, right? So when the fat is broken, it will lead to what? It will lead to that fatty acids. So what are we doing? What are we doing? We are actually, these are the same resources. So it means when we are doing, say, protein and or the fat, they are broken. Practically speaking, they are generating the raw products, raw material for the gluconeogenesis, right? So this is like fat, something which happens in adipose tissues. So this is the reason that when under stress, you'll find that even the muscle weakness, it comes because this is protein, fat, they are breaking and they are giving the raw material, right? This is acting like a raw material for the process of gluconeogenesis. The final result is what? Blood glucose level. Blood glucose rises, right? And then we are ready. Come on, let's fight, right? So that's what really happens. True. Finally, the zona reticularis, right? Let's take it on a new page. Zona reticularis. Matter of just minutes to understand it. But the end result is very interesting. Very, very interesting. What it will make? It will make androgens, right? It will make androgens. And these androgens, they are like sex hormones. Now, the end of this topic, right, which is just within 2-3 minutes, it is vital. It is vital when we'll be dealing with some of the topics of medicine, when we'll be dealing with not medicine, in fact, pathology, right, at that point. Okay, so you remember this and this, right? Just in that zona fasciculata, right? We drew this thing, and that is right. Let me write zona fasciculata. I said just remember these names because we'll be utilizing it. So that was what? 17, right? Those long name. 17 alpha hydroxy. My ghrelin said something. Grrr. So 17 alpha hydroxy pregnenolone, right? That was the thing. 17 alpha hydroxypregnenolone, and one more 17 alpha, and entire thing same, hydroxy progesterone, right? Progesterone. So these are the two raw materials now will be coming directly from zona fasciculata, and they both will be converted, right? They both will be converted by just again just one enzyme, and that enzyme is 1720 lyase, right? This is a new name, so do remember. So 1720 lyase, it will generate two things. One, now again a tricky name is coming up, so we'll write it in our way, right? See, this is 17 alpha hydroxy progesterone, right? So 17 alpha hydroxy progesterone, in last there is one, right? And over here it is lone, right? We'll not consider that it is one, right? It is lone, and this is the pure one, okay? So this 17 hydroxy progesterone, it gets converted by this lyase into andros number 10 AD1. Its actual name would be androstenedione. So over here if we write 10 and over here if we write 1, it becomes a bit easy to remember, right? That's how what I felt. Now if there is any new idea, so you just let me know, but if we write it like this, it becomes easily digestible. And this above one, right, this pregnenolone, gets converted to DHEA, right? Such a long name, but well, D is di, that is easy. H is hydro, E is epi, and A is androsterone, right? Androsterone. So this is dihydroepiandrosterone, and this first one is androsterone tenedione, right? And then once again this is the most enthusiastic hormone, enzyme, 3-beta HSD, right? He'll simply jump and will convert DHEA into the androsterone, right? So over here, this is 3-beta, H is hydroxy, S is steroid, and D is dehydrogenase, right? Writing again, it is hydroxy steroid dehydrogenase, right? Dehydrogenase, right? And it will convert it into androstenedione. So this is androstenedione. Finally, right? There is 17 beta, again HSD, right? So its names are similar. So if you'll draw these things 2-3 times, you will be able to, and the final result has come, the testosterone. Testosterone was done. The process is complete. So actually, here just there are 2-3 names only. And this testosterone will be acting, right? Now, it is possible, it is even possible that this DHEA, it can also be converted directly to testosterone, right? To make things easy, I'm writing it on another page. In a way that we'll be trying to write it in as short as possible, so you'll be able to decipher that newer one, and there we'll change the color. So here it is 17 alpha hydroxy, right? Also it will be a sort of revision. So pregnenolone, right? This was one, and then there was 17 alpha hydroxy and the progesterone, correct? Progesterone. This was converted to DHEA, and this was converted to that our special name andros 1081. So that is andros tenedione, right? Now you know it. So this is done, and this DHEA was getting converted to andros tenedione by this 3 beta HSD. Very enthusiastic, right? So this much we talked about, and then this androstenedione was getting converted to testosterone by this was 3 beta, so this was 17 beta, right? 17 beta HSD. So where is new? This is the same thing what we already talked about, right? But changing the color. As we said, the DHEA can also be converted to testosterone, right? So this DHEA will be converted by, say, this DHA will be converted to andros 1081, that is androstenedione, right? This is andros 1081. Here it is, there is a small change. It is tenedione, the andros, and this is andros, right? I'll just make a small change like this, right? So over here, instead of DHEA going into andros tenedione, here it is going in the form of, say, andros. So we remove this andros tenedione again by 17 beta HSD, and then it is converted into this, it is converted to this testosterone by 3 beta HSD. But basically, whatever the path is taken, finally this testosterone is created. Now this testosterone will be converted to 17 beta estradiol. Unnecessary, right? Such complicated name, it is so easy, right? This, it is nothing but estrogen. It is estrogen. So that's what it is. This is the final thing, right? And here, say estrogen, right? So this is a feminine hormone, right? So let's add some perfume to this. This is aromatase, right? So we added a perfume, right? So final point, that this is how these two sex hormones are created into the adrenals. But say in case of males, this testosterone, when it is created, right? So it is created by testis in large quantity. Adrenals, so it is in very less quantity, right? Similarly, in case of females, say estrogen, it is created by ovaries in large amount. Adrenals, so that is very less. But this is the reason behind that when it is said that in every male there is testosterone and estrogen. In every female, there is testosterone and estrogen. This is the reason. Because testes, they'll be forming pure testosterone. Ovaries, they'll be forming pure estrogen. So then in females, why sometimes there is hirsutism? Hirsutism means those excessive hairs. Why do they come? This is the thing. This is from where it is coming. So actually, because of this testosterone in testis, it is producing more male hormones. So that's why the secondary sex characteristic, that is those hairs, that is in the form of facial hair, right, then axillary hair, then pubic hairs, right, they all are, then hoarseness of voice, heavy voice, all those things, they are forming because of this testosterone. In case of females, because the quantity of the testosterone is very less, so that's why those characteristic, that is heavy voice, etc., they won't appear, right? Facial hairs, they won't appear, but that axillary hair and the pubic hair, right, that is happening all because of that small quantity of that testosterone, right? So that is how this entire system is working, right? So this was our today's session, right? Now I'm turning the chat on, right, just for few minutes, because we have overshot by about, say, how many, 10 minutes, but the topic was a bit lengthy. So that's why, in the initial days, I said that there would be at least one day, then when we need some more time, right? So now the chat is on. And any query, any problem, right? You can definitely just go through this entire, I'll be saving this PDF into our shared folder, right? And any problem, any query, right? Either you put it into chat mode or you send a mail, no problem. What happened? Why it is? Yeah. Chat is not appearing. I don't know why. I'm unable to see it. No worries. No worries. Right. So thank you so much. And any of the query, just let me know. And we meet tomorrow, same time, 7:30. Right. Thank you. Bye bye and good night. See you.